And we're back in the beautiful city of Chicago for the SNMMI 2023 annual meeting, the premier educational, scientific, research, and networking event in nuclear medicine and molecular imaging. The four-day event is well underway and will provide physicians, technologists, pharmacists, laboratory professionals, and scientists with an in-depth view of the latest research and development in the field, as well as providing insights into practical applications for the clinic. SNMMI TV starts right now. Hello and welcome to SNMMI TV. I'm Dina Baer, and I'm here to share all the latest at this year's SNMMI annual meeting. Stay tuned, as we'll be having one-on-one conversations with leaders in the field, highlighting organizations that are at the forefront of nuclear medicine and molecular imaging. Neuroinflammation is a hallmark of many central nervous system diseases, including Alzheimer's, multiple sclerosis, and stroke. Unfortunately, most previously investigated biomarkers for quantifying neuroinflammation in vivo using PET imaging have several limitations. Mausam Kalita, a senior scientist from Stanford, tells us about how they have successfully synthesized and are currently evaluating a novel PET tracer for the measurement of GPR84 expression. Let's see what he had to say and what this means for patients.
The focus of my talk will encompass three different points: identification of a new biomarker of neuroinflammation, and radiosynthesis of a carbon-11 tracer that target this biomarker specifically, and finally application of this tracer in LPS-induced neuroinflammation. We have developed this new tracer that can target a very promising biomarker of neuroinflammation, which is specific to myeloid cells. So we hope that now we will have much better sensitive and specific tracer. What I want people to take home is we have tracers, radio tracers that can detect and target adaptive immune system, but there is serious lack of tools that can address the innate immune activation. And this new tool can detect and target innate immune activation. So basically we have added a new tool in the toolbox. We hope to apply this tracer in more models specific to central nervous system specific diseases, for example, multiple sclerosis and Alzheimer's disease. And also, we hope that we can deploy this tracer in monitoring therapeutic outcomes of immunomodulatory drugs.
We're discussing application-specific PET systems with Dr. Taiga Yamaya. Thank you for being here.
Thank you.
And I know you're giving a talk about sort of expanding the potential of PET in nuclear medicine. And I think before we discuss that, we have to talk about what the limitations and challenges are currently.
The most challenging thing is in detector development. So there are many studies which are improving spatial resolution and timing resolution of detectors, but still the detectability of detectors is an issue. And in my research, we developed a new detector for application-specific PET systems.
And so how do you take that knowledge and then sort of expand the potential for nuclear medicine and PET scans?
Okay, so due to limitation of detectors, conventional PET scanners are all large cylindrical shape. So because of that, so it is preventing to extract the performance of PET scanner, and also it is increasing equipment cost. And with my detector technology, so it is possible to make PET systems as small as compact, as well as improving performance. So I believe we can improve diagnostic performance of PET scanner, and also we can reduce production cost to deliver such good technology to everyone who needs it.
So you make it easier, you make it
Yeah.
less expensive.
That's right.
Clearly more people can use it. It's certainly inspirational and exciting, I'm sure, to the people that are going to hear your talk. What do you hope they take away from it?
A take-home message for myself will be application-specific test systems can expand potential of nuclear medicine. And I believe developing such innovative tools and delivering such things to nuclear medicine physicians, then I believe physicians can save more patient life, and also physicians can improve healthy longevity.
And isn't that the goal, right? Applying the research to the people to make a difference.
Yeah.
Thank you so much for all
Thank
you're
you very
doing.
much. Yeah.
Radionuclide therapies are sparking a revolution in nuclear medicine and are changing the way we treat cancer. AdvanCell is a radiopharmaceutical company focused on the development and commercialization of targeted alpha therapies. Let's take a closer look at their work.
Radionuclide therapy, which bring radiation into the tumor cells to kill tumor cells, has really changed the way we approach the treatment of patients with metastatic disease, both in neuroendocrine tumors and prostate cancer.
Although we are both improving survival, so longevity, and improving quality of life, eventually tumor is coming back.
Targeted alpha therapies are the next step. This is how radionuclide therapy evolves. When you look at the periodic table, it really starts to become clear that lead-212 has the ideal set of physical characteristics for a targeted radionuclide therapy.
We have a pipeline of therapies. Our most advanced is our prostate cancer agent, but we are extremely excited about progressing these other molecules for other hard-to-treat cancers which are currently completely underserved, like metastatic melanoma and pancreatic cancer and colorectal cancer.
Changing the way we treat cancer with targeted alpha therapies is the most exciting thing I've ever been involved in.
Personalizing cancer care demands the right tools in order to get patients the right treatment at the right time. Siemens Healthineers, integrated value chain of precision oncology tools, equips theranostics programs with state-of-the-art solutions at every step of the theranostics care pathway. Let's take a closer look at the incredible work being done at Siemens Healthineers.
We're excited about the field of nuclear medicine and where it's headed, and the unique role Siemens Healthineers is playing to shape the field. Siemens has been developing innovations with theranostics in mind, namely the fastest PET-CT scanners, quantitative SPECT, and increasing patient comfort with flow motion.
At Siemens Healthineers, we provide solutions across the entire theranostics care pathway, and we're really focusing on our precision oncology tools, such as PETNet, lab diagnostics, molecular imaging, software solutions, and therapy solutions. The goal is to fine-tune and help customers with each of these tools to customize their theranostics program. I want to talk to you about John's case specifically and how we utilize PET-CT imaging to select him for his treatment. For him, he has prostate cancer, and here you could see the amount of disease that this patient has. And it's really incredible when you start scanning post-treatment, and you can now use SPECT-CT to be able to see how in his sternum it's just disappearing. And these scans are fairly quick, they're only 15 minutes to be able to assess that therapy response. I want to showcase how our tools, we have auto segmentation now, where we can segment the different lesions and the organ, and right here we can actually see the tumor burden decreasing between cycles and really assessing that therapy response.
The Biograph Vision X leverages our Biograph Vision PET-CT platform. This innovation takes that detector electronic assembly and just takes it to the next step. We're always optimizing our technology and looking to improve and enhance its performance. And by looking at the entire imaging chain, we were able to enhance it to achieve this type of performance, this improved 178 picosecond performance. One of the advantages is it enhances small lesion detectability. One of the other aspects, as a customer, you have flexibility to inject lower doses to your patients and to scan faster your acquisitions, and a combination of both. So there's a lot of enhancements and improvements that you can do with time of flight at the clinic.
This year we are celebrating two important milestones. First one is in the Perspective first anniversary. The system has been redefining performance with new standards for Aspect CT, including an intelligent imaging experience with MyXam Companion, our proven theranostic solutions, since every single Perspective system is both theranostics and quantification ready. Our smart fit configuration that helps our customers that are transitioning from SPECT only to SPECT CT, and the clinical flexibility and versatility of the system. The second milestone is XSPECT's 10th anniversary. XSPECT was introduced at SNMMI 2013, and by leveraging the CT as the frame of reference for the reconstruction, the image quality is improved, and features such as XSPECT Bone and XSPECT Quant are made possible.
I think theranostics is really changing everything because now it's all really personalized, right? Now we're becoming more specific, and we have to work together, whether it's vendors and physicians and customers, for the community so that we can provide that better patient care and give the patient the best chance to long live life.
We're excited about the field of nuclear medicine and where it's headed in the future, and the unique role Siemens molecular imaging is playing to shape the field.
I'm joined now by Dr. Rich Carson, who is focusing on neurology at this meeting. So I want you to tell me, what are some of the challenges currently?
So one of the exciting areas is the range of novel radiopharmaceuticals that are being used to look at a whole variety of disorders, neuropsychiatric disorders. But that process of taking one of those and translating that from the basic science into clinical practice is quite a long process. One of the parts that makes that really interesting, and that is really exemplified by some of the highlights at this meeting, are those studies where we're using more than one radiopharmaceutical to be able to be much more specific, to be able to distinguish one form or another of a given neurological disorders. Because often the clinical diagnosis is a challenging issue. And that's even more exciting in some of the interesting neurological disorders as we're trying to differentiate Parkinson's and multiple systems atrophy, PSP, et cetera. So I think that's where the multi-tracer imaging is going to be a very exciting area, but it is very challenging.
But it does take the guesswork out, so I'm seeing there are a lot of opportunities here in the future.
Correct. And part of that is that how we develop those nice optimized radiopharmaceuticals. And this is one where we're happy that there are a lot of sites doing this, but it's an area where we'd like to see even more growth for both the development and testing of these new radiopharmaceuticals.
And how do you stimulate that?
So part of that is, where the funding sources come from, and there's a lot of different funding sources there. Some of the basic science work comes from the NIH, certainly being able to do that, but also that clinical translation, and there's where we look often with the pairing of how those pharmaceuticals can be paired, sometimes with the pharmaceutical industry. They've been very instrumental in helping to develop novel ligands when they want to be able to assess how their drugs target the specific sites in the brain. And the development of a PET radiopharmaceutical is one of the best ways to be able to demonstrate the engagement of action. In the morning plenary, Dave Mankoff was talking about that in the cancer role. We've been doing that in the brain for probably 30 years. And it's a very exciting and important area for translation of not just the pharmaceuticals, but also the potent drugs.
It seems like with the research, with the technology, there's a lot to be excited about right now.
That's right. I think another part there is not just the new versions of the radiopharmaceuticals, but a lot of specific areas that are new that we've been waiting for a long time. One of the really interesting areas coming out of Yokohama City University are specific tracers for the AMPA receptors. That's one of the main subtypes of glutamates, and they're able to show a really wide range of applications in different neuropsychiatric areas, try to differentiate depression, schizophrenia, et cetera. And this is one that's going to see blossoming in a wide growth. It's a target that's important in these disorders, but often, again, multi-tracer is going to be the way to be looking at that. We look at something like schizophrenia where you may be looking at AMPA, but you'll also be looking at another tracer. You may be looking in the dopamine system, or you'll be looking in the serotonin system in other targets. And I think that's going to let us both differentiate our different sub-patient populations, but also help with the targeted therapies just to be able to look at progression and control of the disease.
And given the mental health crisis in this country right now, it feels like this could be really transformative.
Absolutely, and certainly when you look at opioid disorders, at all of the abuse in that area, looking at a lot of different interesting mechanisms that are associated, some of them very specific to those targets, but also secondary ones. Neuroinflammation is an area that transcends a lot of different diagnostic categories there, and it's very, very relevant in the drug abuse disorders to see the effects of these long-term overdoses of drugs into what's happening in the brain.
Yeah, and knowledge is power. Thank you so much.
Okay, great, thanks.
Well, that's it for episode 2 of SNMMI TV. We'll be back again tomorrow with more exclusive material at the SNMMI 2023 annual meeting. But don't forget, you can catch our show on televisions throughout the convention center, in your hotel room at the Palmer House Hilton and the Marriott Marquis. We'll see you tomorrow.